All right, so where are biocrusts? Biocrusts are found everywhere in the world where it's hot or dry, basically all the places that vascular plants don't do very well. And as you can see from this global map of the deserts of the world, drylands actually make up a really large proportion of the terrestrial surface. And biocrusts are integral to the function of those ecosystems, and so we think it's very important to focus there. And if that's not enough, biocrusts are actually present in a lot of other habitats where vascular plants do just fine. They're present in a host of habitats from shrublands to even forests. And so where they exist is here in these interspaces. You can see them here. And they're actually still contributing really important ecosystem services in these habitats as well. If you want to know a little bit about biocrusts in forest habitats, you have to talk to Matt's grad student, Chris Ives. He's actually doing some work on restoring biocrust mosses in forests post-fire. Okay, so here's the big problem. Biocrusts are slow growers, and they're also very sensitive to disturbance. And as you can see, there's a whole suite of types of disturbances that are happening on the landscapes, especially in arid systems. Things like grazing and fire that are important to disturbing the soil surface. These disturb biocrusts and remove their functions. Things like fossil fuel exploration, recently oil shale has been a big one that causes big disturbance. And even green energies can disturb habitat for biocrusts. And this is because what happens is when they put in a solar installation, they actually scrape the surface clean. And so they're removing all of those functioning organisms on the soil surface. And then when you add climate change into the equation, you can imagine that restoration efforts could be quite a lot slower, or that we could actually be destroying habitat for biocrusts in that regard. Okay, so another big problem when you destroy biocrusts is that you get a lot of dust emissions. So does anybody recognize this picture from Phoenix? This is a haboob. I've always wanted to say that in a talk. I just achieved a goal today, so that's fun. But so when you destroy the surface soil structure, you get airborne dust. That can be major storms, and this can cause health issues. This can cause impact on economy, and certainly can be a safety issue. From an ecological perspective, dust can be a big problem as well. If you think about dust landing on snow, you're changing the color of the surface. You're causing albedo to change, and so you're actually can lead to snow melt happening much quicker. And what happens is the snow, not all of the snow moisture makes it into the soil. It actually sublimates. And so there was a recent study that showed, that this can lead to 5% decrease in water delivery to watersheds. Now that doesn't seem like a whole lot, but if you think about the Colorado River here, it's already over-allocated. And so if you take 5% of the allocation away, we're going to not have enough water to go around. Okay, so let's meet these guys. Aren't they cute? They're very charismatic. They're very small, but they're beautiful. I've listed up here a whole bunch of synonyms. We like to call them biocrusts because it's simple and easy to say, but you've probably seen a lot of these different terms listed before. I'm sorry, I'm standing right in front of you. So the major players here are the cyanobacteria, the bryophytes, these lichens up top here, the cyanobacteria here and here, and then all these bryophytes. So they can come together in all different organizations. It depends on the ecosystem. It depends on the climate. They also can have a whole suite of other guys involved. And when you get this consortium together, this community supports a very vibrant soil food web. And so that can be really key. Okay, so what I want to do is go through building a biocrust and show you the different parts and who they are and how important they are. I'm going to look at this. It's hard to look up there. Okay, so these are the soil stabilizers. They're the primary colonizers. They come in really early in succession. They're green filamentous cyanobacteria. These are their filaments. These are single-celled combinations of photosynthetic cyanobacteria. What they do is they aggregate into these bundles. So they make this really sticky polysaccharide sheath. And you can see on the soil surface here, they're actually holding soil together right on the surface of it. But over here, look at this. They're giant lifters. They're lifting these giant boulders of sand. It's pretty exciting. So anyway, these guys are primary producers. They fix carbon. And they can aggregate the soil in a really good way early on in succession. The next group we like to think about is the stress tolerators. These are also cyanobacteria, but you can see they're dark and they live right on the soil surface. And that's because of these pigments you can see in these pictures here. They actually create their own sunscreens, which is awesome. And so they can live on the surface, they can tolerate desiccation, and they can photosynthesize when conditions are right. They also play a role in stabilizing the soil surface. And when you put these two together, this is just a great consortium of activity here. They're just happy. So these guys stabilize the surface so we can get the dark cyanos up on the surface. And then these guys are protecting these guys from the UV radiation. So it's a nice symbiosis happening there. The next group that we really like to focus on in our lab are the mosses, and we call them the water harvesters because these guys are just incredible. When conditions are right, they take up the water and they can hold 14 times their body weight in water. So you can imagine how good they are at stopping runoff and holding soil in place and feeding the other organisms associated with them with water. So these guys are also photosynthesizers, and they're little plants. They actually contribute organic matter to the soil. The last group are the lichens. These guys really provide the armor to the Biocrust. What's cool about them is that they're probably the best possible erosion protection, and because they're a symbiosis of a fungus, which is the body that protects the photobiont, which can be either an algae or a cyanobacterium, they make a nice package where they're holding the soil in place, they're protecting each other, and they're feeding each other as well. And so one other nice thing about these guys is that the photobionts tend to be some of the best end fixers around. So if we get all these guys together, we have a really nice functioning soil crust. So I want to show you just a few of the ecosystem functions that these guys provide. The first of which is just so fun. They're amazing desiccation tolerators. All of these organisms can tolerate really dry conditions for an indefinite period of time and then come back to life. Mosses are some of our favorite, and I apologize to those of you who have seen this video before, but it's awesome. Okay, so this is a dried up, pretty much dead moss doing absolutely nothing. And then within seconds, I'm going to show you how it can turn into that. So this is some work done by Kyle Doherty. He was a master student in Matt's lab. And he says, moss armor. So watch this. Seconds, they don't have bases that are Okay, amazing water harvesters, and they can activate as needed. Oh, there we go. Okay, so I've already shown you soil aggregation, but I think this is a nice illustration of how they aggregate soil. So what we're looking at here, you can see these petri dishes. What happened here is somebody took a petri dish and cookie cuttered away at the Biocrust. At this edge right here, that's a light cyanobacteria. And you can see how it's really holding that matrix of soil together and giving it structure. So this is aggregating the top millimeters of soil together, and this is just in an early successional stage, so it's pretty remarkable. The next thing that they do really, really well is they add to soil fertility. So you can see carbon and nitrogen going in over here, and that's because if you look at this nice mature biocrust right here, we've got mosses, dark cyanobacteria, lichens, and all of these guys are fixing carbon. They're all photosynthesizing. And moreover, many of these guys, the photobionts and the dark cyanobacteria, the dark cyanos and the photobionts within the lichens are also fixing nitrogen. And so what we get are these little islands of fertility that can support the soil. Food web, and even things like this tiny little plant here, they can get a foothold in there and start to grow because they've got nutrients that they need and stability. Okay, another important role that biocrusts play is hydrology. So I've already shown you how well mosses can harvest water and hold on to it. What we also see with biocrusts is that we change the surface. So we get these really bumpy areas that increase the surface area. And as a result, what happens is that we slow the path of water and we increase the surface area where water can absorb. So there's less runoff generated and more water absorbed into the soil surface. Okay, so my work is focused on drylands of the west, and I'll tell you why in a minute. But if you just look at this map right here, you can see that we are predominantly arid lands, and so biocrusts play an important role here in our region of the world. Moreover, there's a lot of managed lands in the west, so all of these different colors that you can see on this map are federally managed lands. what we're focusing on is really DOD lands, which makes up about 21 million acres of land. But you can imagine that some of the things that we do on managed lands can cause soil disruption. So DOD is interested in restoring soils, and the reason that they're interested in restoring soils is because there's communities near their bases, and they don't like the dust that comes off, and they don't like that they're damaging the soils. And so they put out a call for applications, and the team that I'm working with got a grant from CERTIP, which is Strategic Environmental Planning, Research, and Development, I think. I have to go through it every time. I never know what it's called. Anyway, so they put out a call for soils restoration, and we thought biocrusts are the key. And that's because if we recover biocrusts, sorry, I should tell you, I sometimes abbreviate them as BSE, biological soil crusts. We can restore the ecosystem functions of interest, and we can really reduce dust emission. Okay, so I want to start with this model that is always hard to talk through, but it's really a great model. So this was from a paper that Matt put out in 2007, and it's really a nice review, and I think the graduate students read this one, yeah? So this will be easy for you guys. So when you're thinking about restoration, and especially restoring soils, there's some barriers that you have to address. So at the low end down here, we can think about what can we do passively without really doing much at all, without putting on treatments to rehabilitate biocrusts. And so we can look at these little blue polygons as barriers to success. And so the first barrier is just time. And passively, to achieve the goal of removing this barrier, all we have to do is remove the stress. So for example, in a system that's not severely degraded, there's still propagules present, the soils aren't actively eroding, but the biocrusts are damaged. If we just give it time and we take the cattle off the land, for example, it can rest and recover. So as we move up, we get into active disturbance, and the degree of difficulty in restoring gets more difficult. Okay, so the first barrier we can think about is, okay, so we've degraded the landscape to the point that there are no biocrust propagules on the landscape. So we can't passively just stand by and remove the stress because there's no inoculum there to fix the problem. And so one thing we can do is just add biocrust, and hopefully that will take care of the problem and start restoration on the right path. Another barrier that's a little bit more difficult is what if there's resource limitations? And this can take all types of different forms. You can imagine that micronutrients might be limited, water might be limited. There might not be good microsites for biocrusts to get established. And so this is where you actually have to start thinking about what are the problems on the ground and try to figure out what you can restore. The next level up would be, what do you do if you have actively eroding soils? So the first thing that you have to do to restore this, or remove this barrier, is to stabilize the soil somehow. And what I want you to think about is that this is hierarchical. So if you just remove a stress, but you don't have inoculum, then you're not going to get restoration. If you have actively eroding soils and you just add propagules, those propagules are going to get washed away with the next storm event, right? Because the soils are actively eroding. So we have to think about which barrier is the highest level that we have to achieve to make things work. Make sense? Okay, good. All right, so here we go. So here are the goals of the project that I've been working on through DOD. I'm going to talk you through a few of these in detail. Most of them I'm just going to tell you what we're planning to do. The first one is establish a biocrust nursery, and this is spearheaded by a group at ASU. Sergio's team is here today, and they're actually harvesting their biocrust nursery from Hill Air Force Base sites. So they're creating the inoculum so that they can take it out into the field and use it as an inoculum source, rather than taking biocrust off the landscape and causing a secondary disturbance to correct a first disturbance. The next thing, and this is one of the big major thrusts of my work, is identifying the best methods for restoring biocrusts in the field. What do they need to survive, and is it the same in all ecosystems? Another thing that we're really interested in doing is trying to figure out what are the interactions between soil and plants in terms of biocrust restoration. There hasn't been a lot of work done in this area, but you can imagine that if you add biocrusts and they start to establish, they could have an effect on plants and vice versa. And so we're starting to test some of that out. The next piece after this, which I don't have a bullet for, is to take what we've learned from these steps and put them together into some large-scale studies where we can look at the ecosystem ramifications and understand what happens when we restore biocrusts and plants. And finally, the last goal, and probably the most important, is this communication piece. once we know what to do, we can make recommendations to the land managers and help them to make the right decisions. Our goal is to come up with an easy, cost-effective method that anybody can do. And so we can give them some instructions, and they can go out on the landscape and actually do what needs to be done. Okay, so here's my study system. We're working in a cold desert, a hot desert. So we're working up north at Salt Lake City in the near Salt Lake City in the Great Basin Desert. So we're working on the Utah Test and Training Range at Hill Air Force Base. And then our hot desert comparison is the Chihuahuan Desert. And so this is down in Las Cruces, New Mexico. We are actually partnering with DOD here, but the site that we plan to work on is Fort Bliss, and they're a very active base. And so Jornada is right next door. It's the same ecosystem, and they're set up to do research. And so we're just going to share our findings from this experimental range. Okay, so just a little comparison. You can see that when things are hot and dry at Hill Air Force Base, we have cool, moist springs, hot, dry summers, cold, dry falls and winters, compared to Jornada, which is a warm, dry spring, hot and wet fall, and a cool, dry winter. Most of the moisture happens in the summer, as you can see here. In terms of actual precipitation, they're pretty similar, but the difference here is that at the Great Basin Desert, they get moisture more evenly distributed over the course of the year, whereas the Chihuahuan Desert is very much a monsoonal system. They both get pretty cold. These are mean maximum temperatures. In January, it's 3 degrees Celsius on average in the Great Basin Desert, compared to 14, a little bit more pleasant down in the Chihuahuan Desert. And they both get very hot in the summer. And then we targeted 2 soil types. The idea here is that biocrusts grow differently on different soil textures. And so it might mean that we need different restoration techniques if we're on a coarse soil versus a clay soil or a very fine textured soil. Okay, so here are some data on the amount of biomass in the soil. So this is chlorophyll A. This is just a nice proxy for biomass of biocrust in the soil. So these 2 bars here are Hill Air Force Base, and you can see that they're substantially larger than the bars at Jornada. And so what we can say there is that there's more robust crusts and more biomass of biocrust at Hill than at Jornada. Okay, so I'm going to talk you through some experiments that we ran at Hill Air Force Base. What we did is we went up in April of we went up in April a couple years ago. And after 14 months, we went out and took a bunch of measurements to understand what was happening with the biocrust on the ground after that period of time. So the first experiment that we initiated was to target how much inoculation do we need to add if propagules are scarce. And so in this experiment, what we did is we scraped plots. And so we took all the biocrust off, and then we crumbled it up into small little pea-sized fragments. And then we added it back to these plots in this way. None at all, 10%, 20%, or 40% of the surface area. And then after 14 months, here's what we found. So on the x-axis here, we're looking at inoculation level. On the y-axis, we're looking at biocrust cover. And the 2 different colored bars, the open bars are clay site, and the closed bars are the more sandy textured soil. Okay? And so what you can see is in the controls, we actually got quite a lot of recruitment. And so that's pretty marvelous just on its own. It could mean that we could just passively wait and let the site restore itself. But the best bang for a buck here is at the 10% level. So this is where we get the most biocrust development at both sites. And that's actually nice. It means we don't need a lot of inoculum to get going. Okay, so the next experiment was microsite engineering. So what we wanted to do here is think about some things that might impede the ability of biocrust to really establish and get moving in a meaningful way. And so we created this full factorial experiment where the first thing that we did was scrape the plots, and then we either added inoculum or didn't, and we added these all at the high level because we didn't know what was going to happen. We added a surface roughening treatment, and the reason for this is that there's good evidence that biocrusts establish differently in different aspects, and even, what's the word? Anyway, we can create some microhabitat here to help these guys establish well. Another thing we did is created some shades, so half of the plots got shaded. This is 50% shade cloth, and the idea here is that we can reduce UV damage. we can also reduce the temperatures and potentially reduce evaporation. So this could be a nice cushy environment for Biocrust to grow. The final thing that we did was add water or not. And so this was a one-time event where we added water right after we inoculated. And so this was enough to saturate the surface. And the idea here is that if we can water the Biocrust in, they get a chance to start growing just a little bit and get their feet into the ground a little bit and establish on the surface so that when the next wind blows, they don't get blown away with it. Okay, so this would be a 32-bar graph. So I'm just going to give you the upshot. Here you go. So here are the treatments if you want to look at it. You can see that soil was an important factor. We had better recruitment at the clay site than the sand site. Also, adding inoculum had a big impact. That's good. But so at Burr Buttercup, which is the clay site, it's named that because there are little yellow flowers everywhere, and they have little burrs on them. You can see that the shade plots look like this. They have a lot of dark cyano starting to recruit in there. And this was true of just about any combination with shade as long as we inoculated. So shades were a good benefit at this clay site. At the sandy loam site, the same is not true. Here's a shaded plot that was just completely destroyed. What we found is that the sandy site, little rodents like to move in there. They thought it was great habitat, and they were just so happy that they destroyed our plots. However, when we added all combos together, that was not the case. I don't know why that is, but we did find a combination that works there. So we could do all combinations to get some success. And I think that the takeaway from this is just that you have to pay attention to the site. It can be really nuanced. Okay, so the third experiment we wanted to address was artificial soil stabilization. There's a lot of research that's been done on this, and so we wanted to try some things that had been done already and look at not only do we stabilize the soil, but can we do things that would promote Biocrust at the same time? And so we tried a bunch of things. The first thing that we tried were these polymers, or polyacrylamides. They're called, actually, here are the names. We chose 3. These are fabulous names: Dirt Glue, Terralock, and Soil Tack. Don't you guys want to work with those? Anyway, they are all biodegradable and UV degradable, and so they're supposed to be harmless in the environment. We chose them because they're widely used by the military. They use them on things like landing strips. They use them to stabilize roadsides. And they're pretty effective. You can see how they apply them at different scales. We actually diluted ours quite a lot more than they do. They tend to use a 1:2 ratio. We went 1:8. And actually, I'll tell you, you can still see them on the ground here after 14 months. They're still there. Okay, the second thing that we tried were straw borders. The reason that we wanted to try these, basically what you do is you take a plot and you insert straw vertically around the edges. There's been some really excellent work done in China where they use straw checkerboards. So these are a matrix of straw plots lined up all together to stabilize dunes. And what they found is that the dunes stopped moving. These things worked really well at keeping the dunes in place. And as a result of stabilizing that soil surface, they got really great Biocrust growth on the surface. And then so the last thing that we tried is sort of our everything bagel. We didn't know what the best thing was going to be. And so we thought, a priori, without having any knowledge, let's just try everything. And so what we did was our best habitat modifications, so surface roughening with shade, and then we added a polymer for the stability factor, and we did these with and without inoculation. And so again, I'm going to give you an upshot. It's a lot to take in, and it's nuanced data. But so at the 2 sites, we saw some differences in how Biocrust responded to these stability treatments. We did actually see really great results when we looked at the stability itself. We saw that the soils were becoming more stable with many of these treatments. And they're actually the same ones that did the best with Biocrusts. And so up here at Burr Buttercup, again, we're looking at a couple of plots, one that's treated with a polymer and one that's treated with an everything bagel. And you can see that there's some nice Biocrust development there. Down here at the Sandy Loam site, we also saw some positives. One of the polymers was really great, but inoculation alone was just the best. Seems like at this site, you just add crust and you do better. Okay, so I want to take just a minute to look at the straw plots because we think this might really be a winner. So we actually set up just control plots long ago, and then we realized, we need to have inoculated plots too because the Biocrusts were recruiting so well within the straw plots, and so we set up new ones. These are actually pictures of old ones. And it's hard to see here, but when you poke them, you will find that there's light cyano over the whole surface. Down here, these are actually newer plots because we wanted to try out inoculating them and wanted to see how that worked. And this is only 6 months in, but there's pretty remarkable recovery already, so we think this might be a winner. We're just waiting to get some more data to decide. Okay, so that was a lot. So what I want to do is just give you a quick little recap. So the first study, we looked at adding inoculum, and what we saw is that even low levels, we saw really great recovery. The second study, looking at habitat modification, showed that there can be some benefits, but you have to pay attention to the site. Depending on which organisms are there, depending on what habitat is like, you may or may not get success with different habitat modifications. As for soil stability, we saw that these polymers can be beneficial. Not all of them. Some of them were terrible. Some of them were good. But we're really hopeful that this is a good solution and something we can look into in the future. So going back to our restoration scheme here, it seems like at Hill Air Force Base at least, inoculation is the way to go. That's our highest level barrier that we might have to address, at least for now. We could actually probably just passively stand back and watch the Biocrust come back. But if you want to get things going pretty quickly, inoculation is the way to go. So our hot desert site at Jornada, oh, sorry. Here's a little plug for Sergio. This is a harvest, yeah? This is a whole greenhouse devoted to Hill Air Force Base growing biocrusts. So what's awesome is that we can actually, instead of harvesting from the field and causing a secondary disturbance, we can create a biocrust nursery and inoculate damaged plots from this instead. So this is very exciting to see what happens here. Okay, so I want to move south and go to Jornada. I just want to really quickly show you these two bars. This is Fort Bliss sandy site. This is the Jornada clay site. The green one in the middle is our Jornada sandy site. Turns out when we went down to set up our biocrust plots, there's no biocrust to be found. And so in partnership with Fort Bliss, which is actually very much the same ecosystem, the same parent material, the same plant communities, we harvested biocrust from there. You can see what they look like. Okay, so I've only been back from the field for 2 weeks. We planned on setting these plots up in September to coincide with sort of the dampening off of the monsoon, but really still a wet period. And unfortunately, the government shut down. And so we missed our ideal period, and we got set up in November, which meant that our harvest for our 14-month data was in February. That's just because USGS techs couldn't come in January. Anyway, so I want to just tell you just a quick story of what we see in the landscape. And that is after the last 10 years drought, there's starting to be a recovery. Right now, precipitation is starting to get up to normals and in some cases exceeding normals. And what that's looked like has been major ice storms and extreme monsoon events, which means a lot of flooding. And so what that means on the landscape is that we have a lot of overland flow and saltation. It also means that there's plant establishment. And actually at our sandy site that had no biocrust before, I just took some survey data, and it looks like it's on average about 30% on the landscape now. That's remarkable in 14 months that we went from no biocrust to 30% without any activity. So you can also see there's a whole lot of plants here. These two pictures represent our sandy site. Lots of grass that wasn't there before. And this is actually sort of the story of the system. I don't know if you could tell from the picture, but this is our plot right here. Notice where the biocrust is. It's next to the plot. We saw this again and again, and you know, I'm just like, oh my gosh. So these plots completely filled in with sand, and I'm not sure exactly what happened yet, but right next to the plots, either the inoculum that we added got established, or inoculum that was just moving in the landscape because there was so much moisture is establishing. And so I did do surveys next to the plots as well as away from the plots to try to find out what's happening, and I'll let you know. At our clay site, this is more of a lichen-dominated crust, and what we saw here was major overland flow, even though it's quite flat. So this is what our plots look like here. These dark little bumpy things are water-dispersed lichens and dark cyanos that moved with the water. So again, we don't know if there's treatment effects. I'll let you know. I mean, the good news is recovery is happening, and so that's awesome. But I think the upshot for now, until we really have the data analyzed, is this. There's a lot of soil movement, and so we're going to have to do something to stabilize the soils first. if we really want to get active recovery of biocrusts. Okay, so I want to move into the second part of the talk where we talk about growing these guys. The answer to can we grow biocrusts is yes. I already showed you the biocrust nursery. This is actually some culturing efforts of growing cyanobacteria. The efforts here in growing biocrusts have predominantly been with the light cyano and dark cyano elements. There's nothing wrong with that, especially because you can grow them in large vats. What we were thinking, though, is what if we start with a later successional element? Will that help us to move our biocrust development forward faster? And so we focused on biocrust mosses. You can see they're pretty small, they're pretty dark. So they also have a lot of characteristics that might make them the perfect restoration material, and here they are. They're totipotent. It's a great word. What that means is that every piece of a moss can make a new moss. So you can break it up into fragments and inoculate the land and make mosses. They have incredible desiccation tolerance, which is advantageous for long-term dry storage. So what we can do is we can grow them up, we can harvest them, let them dry, and store them potentially indefinitely until they're needed for a restoration project. I've already shown you how great they are at water capture. They also form these great symbioses with end-fixing cyanobacteria and potentially mycorrhizal fungi, and we're going to explore some of that too. We just haven't got there yet. They're tolerant to really nasty soil types, and probably the best thing is that they generally come pre-inoculated with all the other guys that we care about. So the cyanos, the lichens, the dark cyanos, these guys share the same features that the mosses do, but if we can get them all in one fell swoop, why not try? So here's what we developed. We're so nerdy, we called it the Bryotron. So this is a system for experimentally growing biocrust organisms of all types, but we were focusing on mosses in the greenhouse. So Kyle you might recognize, he was a master's student with Matt, he recently graduated, developed this system here. It consists of a whole lot of irrigation and nested pots. So what happens is we have a little tube that goes in here, water comes in, the pot above it has holes, so we can wick up into the soil surface. We use sand to capture the wicking potential. And so what happens is water moves in really rapidly, it saturates the soil surface, and then drains back out. So what we get, the advantages of this are we get a nice saturated soil every time, so we get even watering. The other really big thing in terms of biocrust is that we don't get a surface disturbance like you would get if you watered from above. What else do I want to say about this? So it's set up with 4 different watering lines, but we could change that. But what's nice here is that we have 216 experimental units, or potentially if you want to go smaller, you could do 5 times that. So this is scalable, and you can really do some nice factorial experiments to understand the biology of these guys. Okay, so I want to just talk you through this experiment in brief. This was the first experiment that we ran in Bryotron. We targeted 2 desert mosses, Centrichia ruralis and Centrichia cananervis. They're obviously the same genus. Centrichia ruralis tends to be, this guy tends to be in a little bit less harsh habitat in the desert compared to Centrichia cananervis, so it'll be just right out in the sun. We chose these because they grow readily in biocrust and they grow readily at Hill Air Force Base, which is one of the places we're interested in. We tried a bunch of different water regimes. So they were kept continually hydrated for 5, 4, 3, or 2 days. And then we tried some different fertilizer treatments. So we used a full nutrient suite, very dilute nutrient suite that was given not at all, 1 time at the start, monthly or biweekly. And this is what our inoculated pots look like at the beginning. So we used 5 milliliters of of mosses. Basically what we did is we cleaned them up as best we could. We let them dry slowly, and then we grated them over a 2 mil sieve to get small fragments. And after just 4 months, this is what we saw. I mean, this is just amazing. Okay, so these graphs are showing you on the y-axis, Centrichia cananervis cover or Centrichia ruralis cover. The red line shows where we started. So the x-axis shows you our fertilizer regime, and then they're color-coded by hydration. And so I don't want you to take away all the nitty-gritty details except to say that, 1, we grew mosses, and we actually grew them pretty fast, and a lot of them. and that there's a different recipe depending on which moss you're looking at. What's so amazing about this is that we were able to get a 6-fold increase in our best treatments in only 4 months, which is something that you might expect over a decade or even longer in the field. So that's a pretty remarkable time frame to grow up some mosses. Even cooler than that is this. So this is the same spot or same pot that was photographed once a month for 4 months. You can see its start and its finish. What we're seeing here is a biocrust. We grew up a lot of the organisms that are important besides just the mosses. Most of this cover here is the dark cyanobacteria. It's just awesome. So we can go from a moss propagule and get a full biocrust that's ready to go out in the field for restoration, which is pretty cool. I don't want you to focus in on all the details except to show you that all the players are here. So these are stacked bar graphs with their different treatments in 2 different moss species. Here are the players over here. Centrichia and nostoc. These guys are dark cyanobacteria. They're end fixers, which is fabulous. We also recruited some lichens. And of course, we got some mosses in there and some light cyano as well. We got all the functional groups that we were interested in. And even cooler than that, if you guys weren't already sold, is that we were able to measure, we just did a quick acetylene reduction bioassay of nitrogen fixation. And what we found is that we're fixing ecosystem level relevant amounts of nitrogen in these biocrusts that we grew up from mosses in just 4 months. So we think that's pretty cool. Depending on the species you started with, it was either more related to fertilizer or the prevalence of these dark cyano members, but that's pretty cool. Okay, so we grew mosses and we grew biocrusts. The next question is, what about these guys? What if we start with lichens? And so what we did, is this. We went out and collected a whole bunch of lichens and mosses from the field at Hill Air Force Base, all from the same spot. And poor Jeff and Harlan worked really, really hard getting the inoculum ready for us. We did almost exactly the same thing. We cleaned them up as best we could, let them dry gently, and then ground them up into the crumbled inocula, and then spoon them onto the surface in different treatments. Again, I'll just give you an upshot here. This is so cool. In 5 months, we got a whole pot full of lichen from this. Yes. Okay, so even cooler than that, the one that really did the best is this guy. This is Colima tenax. This is sort of an early-ish colonizer in terms of lichens, and it's a really great end fixer. So we're really happy that this is the guy that really took off, because we can work with that, and we can take it out into the field and use it pretty well. You'll notice that some of the other lichens and some of the mosses didn't grow so well, but that didn't really matter either, because here is what happened. Again, these are stat graphs. On the x-axis, these are our treatments, controls, 2 moss species together, a bunch of lichen species, lichen and moss species together. I color-coded them, so green is moss. Let's see, blue are the algae. The orange colors, these colors are terrible. The orange colors are lichens, and then the browns are the dark cyanos. And so no matter what, even when we killed the lichens or the lichens died back, we still got really great crusts. We got the players that we're interested in getting restoration. I'll give you some data soon here on what the chlorophyll numbers and the nitrogen fixation capabilities look like. Okay, so this is all fine and dandy. We can grow these guys experimentally, but there's a couple more steps that we have to figure out if we want to go to restoration at the field scale. So the first thing we need to do is scale up. So we've been working in these small pots, but what we need to do is develop an inoculum that's at an ecosystem relevant level. So Sergio's had some great success with that, and so we kind of copied him, and we're calling ours the bulkatron. So this is something that Kyle started with some mosses from Rio Mesa, I believe, and he's had great success. There's really great biomass here, and what you can't see is that underneath the soil surface is all the light cyanobacteria that you could want. Okay, so that's great. We can grow what we need for restoration level. The next question is, will it survive in the field? Kind of an important question, right? And so we're working on that too. So what we did is we took the biocrust that we harvested from Bryotron number 1. We kept it in its water treatments, which is sort of a hardening treatment in itself. And then we subjected it to hardening for 21 days in one of 3 different conditions. luxury. We left them in the greenhouse where they'd been grown up in the first place and kept them watered and happy for 24 hours a day. And then the other two, my daughter here, Julia, was very helpful. We went out and we put them out in the sun. And so half of them were treated with shade, half of them were not. And they were all given water that would only keep them hydrated for a couple hours a day. So getting them used to what they would experience in the field. And then we took them out. We made experimental plots, and we'll go out in a month and find out how they survived the winter. So fingers crossed. I'll give you some data on that when we get it. Okay, so just to sum up what I've talked about here, just from the Bryotron, I want to just let you know some of the keys, I think, out of what we've found so far. The first is that we've developed a really nice system that we can find out about the biology and the physiology and the ecology of these Biocrust organisms. And we think it's a great experimental system. And if anybody has ideas, please let us know. The other thing that we've learned is that we can grow functional Biocrust from the top down, and we can do this really, really quickly. We can get the armor lichens, we can get the UV protection, we can get the water harvesters, the nitrogen and carbon fixers, and that's fabulous. So we can actually develop Biocrust really quickly. And so the next steps we need to think about are scaling up to the production level, and what are we doing with field survivorship. And so we're working on those pieces. We're just not done yet. So I want to leave you with one last thought, and that is, where are we going, and what's the big picture in Biocrust restoration? And so this is sort of our R&D pipeline, so to speak. So it's 4 steps. The first step here we've done a lot of already, and that's developing the culture techniques to grow up the Biocrust organisms. The next thing is maximizing field survivorship. We've looked at this with field harvested Biocrust, and we're starting to look at it with greenhouse harvested crusts, but we have a little ways to go there. The third step is where we're just getting started, and that's delivering Biocrust. How do we do this? What mechanisms do we use? It's fine if you want to inoculate a plot, but what if you have an acre, or what if you have 1,000 acres that you need to distribute Biocrust on to get restoration? And so we're thinking about things like hydro seeders or crop dusters. So anyway, we've got some money to try out some things in a couple different ecosystems. We're going to work with the Colorado BLM and MPG Ranch up in Montana to try some of this fun stuff out. And then the final piece is really deploying what we've grown in the field and monitoring the success. So we've done some of that, but we have a ways to go there too. So with that, I just want to tell you this is not a lone effort. There have been a lot of people involved in this process, not the least of which are the undergraduates and the USGS techs that have helped make this happen. So thank you so much for your attention, and I'll take any questions you might have. Whew.